Hi guys, we're going to go through cellular respiration, which is Unit 1, Topic 1, PowerPoint 14, especially for those of you who've been away or if you're having trouble with this. Keep in mind that this is going to be a topic for the student experiment, not for the exam, so you don't have the pressure of having to learn each intricate detail. But if you choose to do your student experiment on a topic around cellular respiration, you're going to need to know a little bit more of the ins and outs of this. So the syllabus also would like you to know this stuff, so let's go through it. All right, so from the syllabus, you need to recall that organisms obtain energy needed to recycle ATP from glucose in the process of cellular respiration. So you need to be able to recognize that glucose is broken down to ATP, and that's called cell respiration. You need to know that cell respiration is an enzyme-controlled series of reactions, and the reactions follow a sequence known as aerobic respiration with 3 phases: glycolysis, the Krebs cycle, and the electron transport chain, and that requires oxygen, hence why we call it aerobic. You also need to be able to summarize these reactions by the chemical equations, which we'll look at throughout the PowerPoint, and then also analyzing multiple modes of energy transfer. So throughout the PowerPoint, there's different diagrams, etc., that show this in different ways. All right, so we've talked about cells requiring energy to do different tasks within the cell. And we've talked about that happening in the mitochondria and the chloroplasts in plants. The sunlight is considered the primary source of energy because the sun is what's needed for photosynthesis, which the plants are going to do, and that's in a PowerPoint coming up. But then us as heterotrophs, we consume plants, we consume other animals, and then we are able to use that energy in the form of glucose, and then our cells are able to turn that glucose into ATP for our cellular processes. So ATP is the key source of energy that can be used by the cell. Glucose is what we take in from our diet, and then our digestive system has to break that into ATP to be usable by the cell. Glucose is too big and complex to just be used by the cell on its own, so the end goal is to make ATP from the glucose. So this is our ATP molecule here on the right of the PowerPoint. The adenosine and the ribose don't really matter so much. They're just kind of the backbone structure of the ATP. However, the high energy phosphates over here are what's the really important part. And you can see where the high energy bond is shown in between the third and the second phosphate. Now that's important because ATP means adenosine triphosphate, which means we have 3 phosphate molecules there. This is its high energy state. And when the cell uses ATP, it's going to break off one of these phosphates and it's going to release the energy in that bond that is holding those 2 phosphates together. So when we break a bond, that releases energy that can be used by the cell. Once we lose that phosphate, we now have 2 phosphates, and we call that ADP, which is adenosine diphosphate. It means we've got 2 phosphates, and this is the low energy form of ATP. And that would then require energy to be replenished again back to ATP. And you're going to see that throughout this process. This is how the ATP-ADP cycle works. So again, over here on the right, we've got ATP, you've got 3 phosphate molecules. When the cell needs energy, it's going to release a phosphate, and that's going to release some energy that can be used by the cell. That leaves us with ADP over here, which just has its 2 phosphates. And rather than breaking down another phosphate, the cell doesn't do that. It's more efficient to replenish the phosphate and re-break that the third phosphate off again. And so the energy from our food then gets used to power a phosphate being re-added to make ATP again. The process of adding phosphate back to a molecule is called phosphorylation. So you will hear that throughout the PowerPoints as well. Now this bit is probably the most confusing bit where people go, what the hell are these and what is their job? And to really understand the role of these energy molecules, you have to sort of look at the chemistry a little bit. If that doesn't really make sense, that's okay. It's just that if you're really kind of confused about it, understanding the chemistry behind it is helpful. So these molecules are intermediate energy molecules. They're not the end goal of aerobic respiration, but they help out the process. So they're called NADH and FADH2. Doesn't matter what their full names are. They're just electron carrying molecules. They help along the process. They have the same roles, so it doesn't even matter which one's which. But they assist in the production of ATP in the very last stage. So it doesn't really make sense until we get to the electron transport chain how they work. But their job is to carry electrons, which are really important in the electron transport chain. Now, just some background chemistry if you are interested. This is a reversible reaction that takes place with the NADH molecule, and the H is simply a hydrogen. And in chemistry, we learn that a hydrogen is generally a positive charge as a H+. But in this process, one of the things that's confused people in the past is the fact that the hydrogens that get added to our NADH here are in the form of a hydride ion. So while the H+ is positive, it actually carries 2 electrons with it, and electrons have a negative charge. So overall, it seems weird because you kind of think if we're adding a hydrogen to something, it should make it more positive. But in fact, because it carries 2 electrons with it, the overall charge on this hydrogen is a +1 and then -2, overall being a -1 charge, which is a little bit confusing because we always think of hydrogen as positive. So down the bottom here again in our little reaction, we can have an NAD+ molecule which has a positive charge to it, and we can add a H+ to it. And as I just explained, normally we would think that that should be NADH2+ because we've got a positive here and a positive here. But as we know, it carries with it 2 electrons. So we now have a positive here, 2 negatives here. So our NADH is a stable molecule. The job of this is really the adding and the removing of the hydrogens, which you'll see is really important in the electron transport chain. But again, if that doesn't make sense, don't stress. This is the chemistry behind the idea. Okay, now coming back to what I was saying about autotrophs and heterotrophs, this just helps us to really link the cycles of respiration and photosynthesis together. So autotrophs are the ones like plants that produce their own energy. They either use the sun energy, they can also use chemical energy, and they're called chemotrophs. But generally our plants and our algae, they're considered producers because they're using the sun to produce sugar in photosynthesis. Heterotrophs are pretty much everything else. Humans, animals, we have to acquire our energy from an outside source, and we break down our food into sugars and produce ATP through cell respiration. Okay, so our autotrophs and heterotrophs work in a lovely cycle where what we are making is what the plants are going to be taking in, and what they produce we're taking in respiration. So with the flow diagram on the right here, if we have light available to our autotrophs, our plants, they're able to use that light energy through photosynthesis to make organic compounds like sugars, and they release oxygen in that process. Us as heterotrophs, as well as other autotrophs, are able to use the oxygen from the air and the sugars from consuming other organisms to do cell respiration. And in that process, we produce carbon dioxide as a waste product and water, which our cells can use. The carbon dioxide goes into the air and our autotrophs can use that again. So it's a lovely cycle where everybody benefits. All right, so we're going to start the process of cell respiration now that you have that background information, and you can refer to Pearson's section 3.5 for all of the details on this. You can see that there's a mitochondria in the background here because the mitochondria is where this is really taking place, the bulk of it anyway. Now, just an important note on the mitochondria, I've given you that handout where it's got the mitochondria on the front with the hand-drawn diagrams. It's important to be familiar with the parts of the mitochondria that are going to help you to understand this a little bit. When we start with glycolysis, it actually happens out here in the cytoplasm or the cytosol. So step 1 is actually happening out here. Step 2 with the Krebs cycle happens inside the mitochondria here, and that's called the mitochondrial matrix It's just the inside of it. And then step 3 happens in the inner mitochondrial membrane. So it's actually the membrane here that is in the folded inner membrane of the mitochondria. So just keep those in mind as we go through. Now, just to highlight this important point, in the syllabus it states that you should only be able to summarize each process in terms of inputs and outputs and how they're related. So what I'm going to go through here is really just that level. You can watch videos on this on YouTube and they will go into much more detail, but that is quite overwhelming and that is beyond the level that you need to know about this, and that's stated in the QCAA. All right, now over here is an overview of cell respiration. So we'll just focus on this diagram now. And you can see the 3 phases are outlined here. You've got glycolysis, the Krebs cycle, and the electron transport chain. Now over here on the left, you can see that glucose is going to be our starting input for this, and that's going to enter into the first stage called glycolysis. And again, you can see that that's happening in the cytoplasm. I've just crossed that out, but you can see that. Now in glycolysis, we're going to make a compound called pyruvic acid, and we're going to get some of our NADH molecules, and that'll make sense later, but you can see where that doesn't come back into the picture until the electron transport chain. From glycolysis, we're going to get some ATP, but we don't get very much. And then our pyruvic acid is able to go into the mitochondria into the Krebs cycle. From the Krebs cycle, we're going to get some more ATP, and we're going to get some more of these electrons in our NADH and FADH. And again, they come back into it here in the electron transport chain. So that's just a general overview of where this is all happening. And there is many enzymes that help out with this. You don't need to know all of that depth, but remember from what we've learned about enzymes, they're going to be helping things to break down or build new things in the process. Okay, so that's just summarizing what I've said again. So you've got the 3 different phases of cellular respiration: glycolysis, the Krebs cycle, and the electron transport chain. Now, this process can happen aerobically with oxygen or anaerobically without oxygen. The difference is that with aerobic respiration, it's able to do all 3 of these processes. It will do glycolysis, Krebs, and the electron transport chain. If we don't have oxygen available, then only glycolysis will happen. These 2 processes will not happen unless there is oxygen available, because you'll see later on where we need oxygen for that final stage. The cell's overall goal is to do all 3 of these stages, because as you'll see as we go through, glycolysis only makes 2 ATP, Krebs makes 2, but the electron transport chain makes 32 to 34 ATP. And so obviously the cell is going to try to undergo all 3 of those stages using oxygen and making a total of 36 to 38 ATP. When we go to anaerobic respiration, this is really a desperation form of energy when our oxygen levels are low. So when our body's under physical stress, exercise, high intensity, and those sorts of things. And that will be a separate, the next PowerPoint that we do. All right, so the overall chemical formula for aerobic respiration is shown here. And this is happening all the time when we're at rest and when we're breathing in normal amounts of oxygen. It's good if you know this chemical formula because it does help you to kind of understand what's happening as we go through the stages. So we've got glucose here on the left, and its formula is C6H12O6. And what that's referring to is how we have our hexagonal 6-carbon sugar unit. So these are little carbons in between here. So you'll often hear me refer to glucose as a 6-carbon ring, and that's our starting compound. Now we're assuming that with aerobic respiration, we're going to have oxygen available. So that's one of the reactants in this formula. And then at the very end of the process, we release carbon dioxide, we release water, and we get our ATP from this. In the next lesson, when we do anaerobic respiration, you're going to see where this part isn't shown. We don't have the oxygen. We still make some ATP, but we also go off and make lactic acid, which is an unwanted byproduct of this, but that is for next lesson. Now, for each individual molecule of glucose, we get 36 to 38 ATP. That number varies a little bit book by book, but the generally accepted number is around the 36 to 38. Okay, step 1, we're going to look at glycolysis individually. So remember, this is happening in the cytosol. This isn't even happening in the mitochondria yet. And the purpose of this is to break down our 6-carbon glucose ring into 2 smaller sugar units called pyruvate or pyruvic acid. As you can see down here, we've literally just gone from 6 carbons to 2 lots of 3. And if we think about what we learnt about cellular transport, the reason for this is because the pyruvates can more easily enter into the mitochondria than the glucose. Now, in this process, we're going to get 2 NADHs forming. In the handouts that I gave you, where I drew it all out for you, I explained the chemistry behind all of this, and that's, again, more detail than what you need to know. But just in case you're thinking to yourself, where did these NADHs come from? It's all to do with when we break down the glucose, rearrange bonds, there's electrons released and things like that. And the gist of it is that you need to just know that we got 2 NADHs from the process. If you want to know more, have a look in the booklet. In that process, we also got 2 ATPs. So we're going to we invest 2 ATPs into this in order to initially break these glucose bonds. And we actually get 4 from the process, but the net gain is 2 ATPs. So if we think about this in terms of inputs and outputs, we're going to have a glucose, and we're going to break that down into 2 pyruvates, and we're also going to get 2 NADH and 2 ATP. I'm going to change my pen color here and be fancy. All right, now if we think about what happens to these products, our pyruvate is now going to go off and enter the Krebs cycle. So it's actually going to go and float on into the mitochondria and start the Krebs cycle. Our NADH that we made is going to leave us for a little bit, and it's going to come back, and you'll see where he reappears, in the electron transport chain. And our 2 ATPs are now available for the cell to use. So it can actually use those 2 ATPs to power things and start using it straight away. So we're not really going to factor that into the equation any further. Okay, now you're going to really find Mr. W to be quite enjoyable if you haven't seen him yet. So I'll let you enjoy that in your own time. I could get out my guitar and try and reenact what he's doing, but nobody needs to see that. All right, so to take us back to where we currently are in the process, we've now looked at this bit in the green circle. So we've looked at glycolysis. In summary, we've got glucose going to pyruvic acid. We have released some ATP for use by the cell, and we have released some NADH, which is going to come back and greet us again in the electron transport chain. Now our pyruvic acid is about to go, as you can see here, and enter into the Krebs cycle. But first there's a little intermediate step that happens here where you can see we're losing some CO2. This part's called the linking reaction. And it's just kind of like an intermediate step between glycolysis and the Krebs cycle. That is now going to be shown in the next slide. Not the next slide. We're going to look at this summary table where we have our glycolysis summary there. So the locations in the cytosol, we used 1 glucose to make 2 pyruvate, 2 ATP, and 2 NADH. And here's our linking reaction. So over here on the right, we've got our 3 carbon pyruvates. So remember, they are 3 lots of carbons, and we have 2 of them, don't forget. And what happens in this step is we actually produce another NADH, and we're going to remove a carbon in the form of CO2. So we're actually going to remove a carbon off our pyruvate, and that's going to join up with some oxygen from the cell, and we're going to exhale that as CO2. That's going to leave us with just 2 carbons remaining, and a little enzyme called coenzyme A is going to bind to this 2 carbon unit, and the 2 carbons are called acetyl, and they join up with this coenzyme A to make this compound called acetyl-CoA. So that's just a 2 carbon molecule attached to an enzyme. That is going to basically make it easier for this to enter the Krebs cycle and go off into the Krebs and electron transport chain. Again, if you want to know the chemistry behind this, have a look at the booklet, but bear in mind that we're doing this twice because we have 2 pyruvates. So in this linking reaction, we're actually getting 2 acetyl-CoAs, 2 NADHs, and 2 CO2s. We'll come back to this point once we finish the Krebs cycle. All right, now I've removed my face from this slide because I need you to be able to see what's going on here. So the whole point of cell respiration is to break down our 6-carbon glucose, remember this fella, and break the carbons down further and further so that we can release little pockets of energy. So we've already split it up into 2 lots of 3, and now we've removed 2 in the linking reaction. So we've got 4 carbons left, and the purpose of the Krebs cycle is to further strip those carbons apart and release energy for use by the cell. So our acetyl-CoA is our starting reactant here. It does 2 laps, and in the process, because there's 2 of them, it's going to lap twice. So we're going to, in the process, release 2 carbon dioxides because we're actually splitting our carbons apart. We're going to get 3 NADHs, 1 ATP, and 1 FAD. Now it repeats that again for the second acetyl-CoA. So we're going to double all of those figures, which you can see over here on the left. So we've got 2 ATP, 1 for each acetyl-CoA, 6 NADHs because we're timesing this by 2, 2 FADHs, and 4 CO2s. Now, when we do the totals for the Krebs cycle, we also have to factor in what we made in the linking reaction, which is technically part of the Krebs cycle. And remember, we got 2 CO2s from the linking reaction, and we got 2 NADHs. So in the summary table shortly, you're going to see where we've got 2 ATP, 8 NADH, 2 FADH, and 6 CO2s, just in case you get confused where those numbers have come from. All right, so back to our summary again. We've made some acetyl-CoA here in the linking reaction. That has to happen first. We've released CO2 from our 2 pyruvates to do that. Acetyl-CoA has entered the Krebs cycle. We've made NADH and FADH2, which are, again, just like our NADHs from glycolysis, they're going to come into the electron transport chain at the end. We've released some more ATP, and we've released some more CO2. So in summary, this takes place in the mitochondrial matrix in the middle of the mitochondria. We put in our 2 pyruvate. Remember, that was changed into 2 acetyl-CoAs, and our overall outputs was 6 CO2s, 2 of those being from the linking reaction and 4 from the Krebs cycle, 2 ATPs in total, 8 NADHs, 2 from the linking reaction and 6 from the Krebs cycle, and then 2 FADH2s. And we're now going to see what's going to happen to those. Remember, this has now been breathed out from the body. This is a waste product in the human body, so we don't keep this or use it in the cell. We exhale it as a waste product. Our ATPs, just like the ATPs from glycolysis, can be used by the cell. They can go off and be used for jobs that the cell needs to do. And like our NADH, these are going to come into the Krebs and these guys are now what's going to be powering this electron transport chain. We no longer have any more carbon sugars. We've broken those down, and now it's all the job of the electron carrying molecules to power the electron transport chain. Again, you've got Mr. W if you would like to go and see his part 2 video on the Krebs cycle. Do yourselves a favor, listen to Mr. W. All right, so now we're looking at the electron transport chain. We've broken down all the glucose. So we now are going to be using those NADHs and FADHs to basically work through this series of integral proteins. Remember these special friends that we learnt about earlier in the term. They're going to be basically, this is in the inner mitochondrial membrane. So if we think about the mitochondria, that's the outer membrane. The inner membrane is the one with all the folds. So if we were to take a chunk out of this, that's the membrane here that we're looking at. This little space in between up here is the gap in between the two membranes, which we call the intermembrane space. All right, now what happens in this stage is basically we've got our 8 NADHs and our 2 FADH2s. And what they do is they work their way through these proteins. And as you can see, they're going to break down from NADH into NAD+. That's obviously removing a hydrogen from the NADH. And the hydrogen is going to go across this membrane into the intermembrane space. As it does that, remember our hydrogen carried with it 2 electrons. So the 2 electrons actually pass through these proteins. At the same time, our FADH2 is being turned into FAD. So it's removing 2 hydrogens, and they're going to pump across into the intermembrane space. The whole purpose of this is we want to establish a really high concentration of H pluses on this side of the membrane to make a really high concentration gradient. In this one here, this is the last stage where the electrons are the electrons get converted into water, which is why we make water as a product of respiration. And you can see here that oxygen is required for that to happen. If we don't have oxygen there, it can't join with the hydrogens and use up those electrons to complete the electron transport chain. Now, the really important part of this is the whole idea of the NADs and FADs was to remove their hydrogens at this stage. We've stripped off their hydrogens and we've pumped them into this space at the top. Heaps and heaps of hydrogen ions. And the really important bit here is this enzyme here called ATP Synthase. It's an enzyme, but its structure is different to the kinds of enzymes we've looked at. It works like a pump, and it basically, hydrogen ions are going to bind to this enzyme and cause the pump to kind of rotate. And when that happens, it facilitates phosphate being added onto ADP. So that's going to replenish our ATP molecules. Hydrogens don't turn into ATP. That's a common misconception. What it is all of the hydrogens that are up here act as the substrates to activate this enzyme and make heaps and heaps of ATP. And we get, remember from this step, 32 to 34 ATPs. Okay, so that's just putting that into words, what I've just explained there. So the membrane proteins carry hydrogen ions across the mitochondrial membrane. The electrons from our NADH and FADH are passed from one protein to the next. We get a really high hydrogen ion buildup on one side of the membrane, and that's going to create a really strong concentration gradient. Remember, molecules don't like to be packed in together like this, and we've learnt about ions not being able to just diffuse back across the cell membrane. So these ions are really keen to pump themselves down this ATP synthase because they want to separate out, even out their concentrations. So they're going to move through the enzyme and we get our ATP. This is a close-up of how the ATP synthase actually works, and if you search this on YouTube, it gives you a nice visual of this. You can see again up here, you've got all these hydrogens built up in the intermembrane space. They actually bind onto the enzyme, and it works by rotating this rotor. You can see here it rotates clockwise when the H plus goes through. And when that happens, it actually activates some sites down here where phosphate comes in and joins to ADP. Remember, that's our low energy molecule, and it adds phosphates to it to make our ATP. There's a summary video that shows this and all of the complexities of what's happening here, and it also points out that these aren't just happening one thing at a time. It's all happening together, and it's kind of a cycle that's always happening in the cells. All right, so in summary now, we've used our NADH and FADH through the electron transport chain, and we've produced bulk ATP from that process. Okay, in summary, that's happened in the mitochondrial membrane. We've had to use oxygen for this, as well as our NADHs and FADHs, and we've made 32 to 34 ATP. Actually, what's also not shown here is that we've made water, and that's the key sort of important part there. Now, remember, we're also making, when we break these down, we're making NAD plus and FAD plus, which then get used again back here to make NADH again, which is kind of that nice cycle between the NAD pluses and the NADHs. They work, sort of what we end up with at the end goes back into the start to repeat the process. There's some duplicate terms that you will come across, especially if you choose to study this for your student experiments. So I've just highlighted them here in case you get confused with what they mean. So the Krebs cycle is also referred to as the TCA cycle, or the tricarboxylic acid cycle, or the citric acid cycle. Don't ask me why. There's some serious complexities around that, but they all refer to the Krebs cycle. Krebs is the dude who discovered this. Pyruvate's also called pyruvic acid, and you would have seen this one in that summary figure earlier. Chemiosmosis is another word for what happens in the electron transport chain. So it's referring to the H plus gradient that's forming across the membrane of the mitochondria. Okay, now these words you may or may not come across, but remember I mentioned phosphorylation is adding phosphates to something. Substrate level phosphorylation is talking about glycolysis and Krebs, so everything except for the electron transport chain, and it's referring to ATP being produced just from ADP, just in the process where we got our 2 from glycolysis, 2 from Krebs. Oxidative phosphorylation is talking about the electron transport chain where we're getting ATP from our NADH and FADH. All right, so have another look at those goals. Remember, you need to recall that this energy comes from, we make ATP from glucose in cell respiration. You need to know that that's controlled by enzymes and that we have glycolysis, Krebs, and electron transfer chain, and that requires oxygen to do those 3. You need to be able to summarize these by the chemical equation and also know those inputs and outputs of each of those phases. And we've also looked at multiple modes of energy transfer through these different diagrams in the PowerPoint. All right, so these are some activities that you might want to do. There's another summary video there that you can have a look at. The activities that I've asked you to do specifically in class are the handouts. So you've got the ATP-ADP cycle handout. You've got the chemical reactions of respiration handout, and that's pretty much it. And then I will also give you an optional handout which gives you heaps and heaps of practice questions.